Okay, hi everyone, I'm Xiaojun Su from Xiaojin. At Xiaojin, we specialize in the nanopore sequencing services and also developed new applications on the ONT platform. So today, I'm excited to share our talk, driving the genome innovation with ONT from ultra-long reads to multi-omics. And in today's talk, I'd like to share how we use the ONT for the T2T genome assembly, the experimental innovations, and several multi-omics approaches. And together, these developments help us explore the genome innovation more effectively. So the first, I will begin with the T2T genome assembly and highlight how our the ONT-only approach. And when we look through the history of the genome assembly, each stage has really followed the improvements in the sequencing technology and the assembly tools. So as T2T assemblies are improved so fast in recent years, we started asking, how do we make this easier and faster across spaces? So that what led us to an ONT-only workflow. And this strategy is a the single platform and still highly accurate. And for the genome assembly, both the ONT and the HiFi long reads are powerful, but they perform differently in the challenging regions. For example, in the GC region or the highly repetitive regions, the ONT maintains stable coverage, whereas the HiFi often drops off. And this stability is exactly what allows us to achieve true T2T assemblies using the ONT data alone. And for the genome scaffolding, we use a Pore-C. It doesn't require the built-in enrichment and the PCR amplification, so the data is less biased and it also can capture the multi-contact interactions rather than just pairwise one. And it can also providing more effective signals for scaffolding. And here we compared the 100x Pore-C and the 20x Pore-C data. We found that in the highly repetitive regions, the Hi-C often loses signals because the short reads mapped and get faded out. But the Pore-C long reads can span these regions, so the international signals stay clear. And besides, the Pore-C also can fix assembly errors caused by the Hi-C. So in this case, a region was incorrectly inverted in the Hi-C scaffolding, but the Pore-C long reads in this region, it clearly showed the mistake and allowing us to correct the inversion and restore the purple chromosome structure. And based on this, here we present the ONT-only T2T assembly workflow. We use the ONT ultra-long reads for the contiguous assembly and the Pore-C data for the chromosome-scale scaffolding. And with the HiFi exam assembler, along with our gap filling and the telomere extension steps, we can generate the complete T2T assembly. And using this ONT-only workflow, we've now assembled nearly 50 different sample types, from the small genomes to much larger mammalian genomes, and the workflow is very efficient. The small genomes just take a couple of hours, and the large one finish much quicker than before. So here is our test results. In our test, for the challenging plant genome, we used the 50x the ONT data with the N50 of 60 kilobits and the 26x Pore-C data. The final genome is over 1 gigabit and fully gap-free across all the chromosomes. It shows that our ONT-only workflow can also handle the large complex genome. And besides, for the mammal genome, we obtained a gap-free assembly using only the 60S ONT data with a reads N50 of 136 kilobits. So such continuity is very difficult to achieve with the previous assembly methods, and showing that the nanopore ultra-long reads can effectively resolve the repetitive regions. And all of these is depending on the wet lab performance, especially to obtaining the ultra-long DNA from the challenging samples. And to obtain the ultra-long DNA, we developed our own extraction kit. It has 3 main steps. The first, we gently isolate the nuclei from the tissue, and then we extract the DNA from the nuclei and check its quality. So importantly, the entire workflow is done manually, and this gives us fine control at each step and allows us to consistently recover the ultra-long DNA stable, suitable for the ONT sequencing. And we also get a very good results from challenging sample types, including the fish testes, the molluscan tissues, and the insect pupa, and complex plant tissues, such as rich in the polysaccharides and the polyphenols, or the highly fibrous tissues. So even with these tough specimens, we consistently obtained the high-quality ultra-long DNA suitable for the ONT sequencing. And here were the actual results from the ultra-long sequencing projects. The real-time sequencing data shows an estimated M50 of over 100 kilobits, with the longest reaching up to 600 kilobits. And here was the cucumber genome case. This is actually a very challenging genome to assemble. Although the genome size is only a bit over 300 megabits, it has an extremely high repeat content. And in previous assemblies, no matter whether we used the ONT or the HiFi data, we always lost almost 1/3 of the genome. And even when the read length reached around 100 to 150 kilobits, the many regions still could not be assembled. So the problem wasn't coverage or the depth. It was simply that the repeats was too long and too complex to be bridged. So in this project, the key improvements was the read length, and we generated the ONT ultra-long reads with an M50 above 200 kilobits, and finally allowed us to span the long repeat blocks and complete the genome assembly. So when we compared the new version 4 assembly with the previous version 3 genome, we found that most of the newly assembled sequence are the repetitive elements, and including the transposable elements and the different types of the DNA repeats. So in particularly, the number of the assembly of the copia and the gypsy elements in the version 4 is nearly doubled compared to the version 3. So the case showed that the assembling highly repetitive regions is really depending on the ultra-long reads, which is something the ONT uniquely provides. And the ultra-long sequencing is only possible when the DNA quality allows it. And so the next challenge is we tackled how to maximize the ONT read length when working with some difficult samples, such as the silica-dried species. And for many rare species, these samples are the only material available, but the DNA is often highly fragmented. So to address this, the core idea is to recover the nuclear efficiently and release the cells very gently, so we can retain the long DNA fragments while removing the containment maintenance. And using this approach, we consistently obtain the DNA around 20 kilobits with good quality and yield. And after the ONT library preparation, the reads M50 typically ranged from 40 to 20 kilobits, and close to what we see with the fresh samples, and making reliable genome assembly with the HiFi exam software possible. And here are our assembly results. And for the 2090 silica-dried samples, the HiFi exam produced a contiguous M50 of 80 megabits. And after ragtag scaffolding, the scaffold M50 reached 22 megabits with a Bosco over 98%, and showing excellent continuity and completeness. And for the 2022 samples gives a contig N50 of 21 megabits, so with scaffolding also performing very well. And this shows that our optimization extraction methods and combined with the hi-fi software, it's worked very well for the producing the high-quality genomes from these challenging samples. So with this optimization, we can now include as many samples as possible for the genome research, whether it's the silica-dried specimens or the high-quality materials. And from the collections through the optimized DNA extraction, the ONT sequencing, and assembly, and then to do the scaffolding. So we can generate the high-quality genomes across a wide range of species, and this greatly broadens the scope of the pangenome research and allows us to include samples that were previously left out. And with these high-quality genomes in hand, we are ready to go beyond the sequence itself and explore the regulation and the functions through the multi-omics. And here we've built a nanopore associate library to study the poly-A tails, and using the dual adapter primers, and we can capture the full-length transcripts from 5' to 3' and measure the poly-A tail lengths accurately. And it also can give us the information we need on the transcript expression and the tail regulation. So in the potato case, under the salt stress, the tail associate reveals the dynamic APA changes, and with some transcripts shortening or the lessening their 3' UTRs. So these changes influence the stress response genes and demonstrating how our methods can uncover the regulatory mechanism controlling adaptive traits and identify the potential targets for the crop improvements. And with the ONT's direct RNA sequencing, we developed a workflow for direct link RNA sequencing, and we remove the rRNA and sequence the RNA directly, and letting us capture almost all the RNA tabs and see their modifications. And we compared the regular DIS with the link DIS. As you can see, the link DIS can detect more the RNA tabs, especially those without the poly-A tails. And besides, it's clear that the linker DIS can capture a broader range of the RNA tabs, and it's also can providing a clear view of the linker RNA diversity and the modification patterns. And beyond the RNA, we pushed the nanopore into the epigenomics research with the SMAC-seq. It's building on a 2020 workflow. The SMAC-seq provides the single molecular chromatin accessibility and the whole genome DNA methylations, and it can give us the long reads with an N50 around 20 kilobits, so all from the same data sets. And this completes the long read toolkits and covering the genomics, transcriptomics, and the epigenomics in one platform. So then I will give a quick summary of our technology. So this is what we mean by all-in ONT with a single nanopore sequencing platform, and we can do it all from the T2T genome assembly to 3D genome research, the epigenome, and the transcriptome analysis. So this gives us a truly integrated view of the genome structure, the chromatin accessibility, the DNA methylations, the full-length isoforms, the poly-A tails, and the RNA modifications. So all from the same technology is from the nanopore technology. And for us, our cell gene team can handle the entire workflow from the extraction to the sequencing, and the T2T genome assembly, the multi-omics integration, and covering every step of a complex project from start to finish. So that's all from us, and thanks for listening. We're excited to share more and discuss collaborations at our booth. Thank you.